I'd like to thank you all for your attention this afternoon. I will first deal with a disclosure that we formed a company, Biocystics, to sustain in the public MPS database through commercialization, and I have equity interest in that company. So today I want to give you a brief background on the MPS database and the challenges that it addresses, a live tour of the MPS database, which will be the bulk of the presentation, and then a little bit on some portals and a time for discussion. So the MPS database was designed and implemented to support the use of a wide range of model types. We have, for example, currently there are models that span from 2D culture models all the way to complicated integrated multi-organ MPS models, and a lot of other kinds of models in between. And so the database really was developed in order to support and compare data from all of these kinds of systems. So another important aspect, and I'll discuss a little bit about iPS cells farther down in the presentation, but there's been an increasing use of iPS cells, and for good reason, because it allows the production of individualized human organ models, as well as to create sets of cohorts of disease models that can be used to as a substitute or as a preclinical trial, for example, in order to improve the operation of clinical trials. So in terms of selecting patients to be involved in the trials and that sort of thing. So we now have this large and growing selection of different kinds of in vitro models that can be used. And one of the challenges associated with that is how to manage all of the data that's generated and then compare models, compare data, compare assays amongst all of those models. So that was the driver behind the microphysiology database. We support all of these model types that I mentioned. We integrate that data from those models with preclinical data and clinical data, as well as data from other databases, including many online databases and proprietary databases. And we use that data then to provide tools to evaluate reproducibility, safety and efficacy, and to build models of biological functions based on these experimental models. So the microphysiology, this is a picture of the homepage of the microphysiology database. And one question is, well, why do we need a microphysiology database? And I think there's three general categories of reasons why it's useful to have the microphysiology database. The first is that it, I think, really facilitates the development and implementation of models by being a resource that has information about the models as well as protocols that were used in the development and implementation of those models. It provides a tool for managing data and especially the metadata from the MPS studies. And so having the metadata is really important, and we've put a lot of effort in making sure that in addition to capturing the data output from the studies, that we capture the study configuration data that's important in terms of the interpretation of the results. And then finally, a variety of tools that are useful for analyzing the data, evaluating reproducibility and comparing reproducibility, not just between individual models, but even between models that were produced at different sites. And we're in the process of developing tools to establish clinical concordance, which we think will be, an important factor in terms of these experimental models becoming mainstream. So to make all this work, we've developed a relatively simple single entry system that's essentially a workflow that walks you through or carries you through the implementation of a MPS project. Starting with the design of the study, the configuration and setting up of all of the chips or plates that go into the study, the analysis and processing of the data that's generated in the study, the evaluation of the study and tools like power analysis that can be used to optimize studies for the next round, and then a means to share all of this information and data amongst either specific collaborator groups or to the scientific community in general. And we have two models for sharing data. We have our public database, in which the data providers get to control when and how the data is shared and to whom it's shared. And then we have a version that's targeted for for-profit companies where they want to keep their data proprietary and they prefer to have the database installed behind their firewalls. And so that was what's behind the formation of Biosystics to support these companies, such as pharmaceutical companies and biotech companies or CROs that want to keep their data in-house and don't necessarily ever intend to make it publicly available. And the commercialization of the MPS database will generate revenues that will support the public MPS database. And so the goal is really to make the public MPS database be a self-sustaining database. So that's all on the background. I want to jump right into the demonstration. And in the demonstration, I just want to introduce you to the overall navigation and workflow. We'll look at some models. We'll look at the components that are used to set up a study, a little bit on how to look at study data and evaluate it, some data analysis, computational modeling, and then a quick tour of a portal, and then we can get on with questions. So this is the URL for the MPS database, and it will take us there. And hopefully you see the MPS database on your screen now. So this is the home page, and the home page is laid out to correlate with that workflow that I showed earlier. So we have study components and essentially study design elements that are in the top row, and then study implementation elements that are in the bottom row. I'm not going to spend a lot of time on this. What I would encourage you to do is go to the help menu. And in the help menu, we have a series of webinars that were presented last year. And these provide a good overview of the MPS database functionality and operations. And so you can learn how to navigate around the MPS database a lot in this first webinar. And so I would encourage you to go take a look at that so that we can move on to the rest of the demonstration. So back to the home page. So what I'm going to first show you is the global search function. I think the global search function is something that isn't really used as much as it could be, and I think it's quite a valuable function. And so, for example, if you're interested in kidney, you can just type kidney into the search function, and it will bring up a list of all things in the database related to the kidney, the cell samples, cell types, devices, drug trial results. We have MPS models and FDA reports, and way down at the bottom, we have a list of all of the kidney-based studies. So there's a lot of information there. It's a lot to scroll through. You can click on this little box and just go right to the studies. if that's your principal interest. You can always also type in, for example, a compound name, like, if I can spell it correctly, gentamicin, and it will, whenever you type into the fields in the MPS database, it will pop up options that you can select from to simplify data entry. So gentamicin, so we hit return, and it'll bring up the compounds, of course, and then some drug trial results, open FDA reports, and then studies in which gentamicin was used. So it's an easy way to find studies, find out if there were studies with a particular compound, and then what those studies are, and be able to look at them. And then the other thing is just, if you're not sure exactly what I mean by a study, you can even just type in a term like that, and it'll bring up the glossary, and it has definitions of all of the study-related characteristics or terminology in the database. So I think one of the things that people are probably interested in is looking at what kinds of MPS models there are on the database. If you're interested in implementing a model, it's always a good place to start, is to say, well, okay, what models are there? In all places in the database where there are tables like this, a quick way to sort through the table is to use this search function. And all I have to do is type in a few characters like KID for kidney, and quickly get a list of all of the kidney-based models. And you can also use the titles of the columns to sort. For example, I can sort by devices, so then I can see all the various kinds of devices that have been used in constructing kidney models, and there's quite a variety. For example, we can look at a typical model, which is a Nordis-based triple channel model, as one example. There are things are, sorry about that. So there are, again, we'll get to the kidney models and look at the devices. So there's 24 well models. We have 96 well model versions. The CN Bio model, which is a 12 well model. Memetis has a 3-lane model that was tested at Texas A&M that has 46 models on a plate. And then Texas A&M also did a 2D version of the kidney, and in a 384 well format. And so you can go from relatively biomimetic models based on chip architectures all the way up to more simplified microplate-based models, and a lot of options in between. And then we also have quite a number of liver models. There's 43 various types. There's gut models, there's brain models, there's bone models, there's muscle models. So lots of different kinds of models that can be evaluated and used in studies. So the other part that goes into creating a study are the study components. And so we have a lot of study components that can be used directly to build studies. An important one are targets, and these are assay targets. And so if you want to run an assay, for example, like KIM1, you can just, again, type in the search function and find that, yes, indeed, we do have an assay for KIM1 that's already in the database. And that assay will also include a method associated with it. We've already talked about models and devices, compounds. So we have a lot of information about compounds in the database that can be useful in designing studies and choosing compounds to use in a study. If we just pick a compound in particular, like say pioglitazone, and we open up the record for pioglitazone, we see there's some information, some links to other resources like clinical trials and drug bank. There's some information about the compound, its compound properties, its drug-like properties, its molecular properties, a brief summary of ADME properties, and then a list of biological targets and the target types, the pharmacological action of the compound on the targets, and the type of action for that particular compound. So that's pretty useful for looking at a single compound. If you are looking for a wider range of compounds or other targets, you can use the bioactivities. I'm not going to take the time to go through that, but that is explained in that first webinar that I mentioned that you can access via the help function. The adverse events table, I think, is quite useful in terms of selecting compounds. And I'm going to go to the compare adverse events table, which allows you to select multiple compounds at the same time and then compare their relative adverse effects. So again, we'll start with typing in kidney. We get all of the kidney-based adverse events ordered by their frequency of reporting. We can pick some of the top most frequent adverse events for the kidney. We get a list of compounds, and there's quite a few compounds that we could choose from. I'll just pick a couple. Maybe we'll look at gentamicin again, and cisplatin, and isoflurane. And then if we scroll down to the bottom, we see for the selected adverse events, we see for each compound its pattern of adverse frequency of adverse events. And these are normalized to the number of reports per 10,000 mentions. And the 10,000 mentions is data that we get from the CDC that we normalize to, and it's data on the frequency of usage of all of these drugs. And so interpreting the number of reports of adverse events isn't as informative as the normalized number. And we can show that by looking at just the raw count of the adverse events for these compounds. And you can see that if you were just looking at the raw numbers from the FAERS database from the FDA, you would see a lot more adverse event reports for cisplatin and gentamicin than for isoflurane. However, isoflurane isn't used very much because of its adverse effects. And so the normalization then takes that into account and gives us a more representative picture of the relative toxicity of these particular compounds. So, and one last item in the study components are the cell samples. And so we have a lot of cell samples that have been used in all of these studies that can be used as a reference for finding cells. If you're looking for cells for kidney studies, there's lists of cells here that have been used in the studies that are in the database, or you can search on liver cells, or if you're looking for iPSCs, you can search on iPS cells, and then you see a list of iPS-derived cells that crosses the organ types. So a lot of different ways to choose cells to use as part of a study. So now that we've talked a lot about I've talked a lot about how to build a study or what goes into building a study, I think it's useful to look at studies. And I'm going to use a couple of liver studies that were run at Texas A&M because I think they're very illustrative of the kinds of things you can do with the MPS database and some of the values of the MPS database. So this is a liver study that was run at Texas A&M where they looked at multiple compound treatments. And So when you open up a study, you come to the study overview page. And the first thing on the top of the page is a table that we call the difference table. And you'll see up here in yellow, it says difference only. And so this table has reduced the configuration of the study, the metadata of the study, down to the variables that were varied in the experiment. And so it gives you a quick way for a study to see sort of what were the variables for that particular study. If you want to see all of what went into the study, you can click up here on all components, and then you get a list of all the components, the various cell types that were used, and the numbers of cells that were used, all of the settings that were used in terms of running the study, the individual chips that were used for those particular studies. But this difference table is really quite nice, because then it shows you exactly amongst these treatment groups that there were variations in the compounds or the compound concentrations that were used in the study, and that we have one control and then the rest, one control group and the rest are treated groups. If you scroll down farther, you can see for each chip the exact configuration, but that's a lot of detail and sometimes useful to look at, but I'm not going to go into that now. The second page to go to that's really useful is the study summary page. And that page shows you a description of the study, what was being tested, or what was the purpose of the study. In most cases, there's a protocol that can be downloaded for the particular study. Some pictorial description of the study, or sometimes it's just a list, for example, here of the compounds that were used in the study. It varies a bit from study to study. The assays that were run as part of the study. And so you can see that there are some sort of classical liver assays looking at albumin secretion, BUN. But also there were some LC-MS measurements looking at the percent recovery for the compounds that were used in the study to better understand whether the compounds were being absorbed at all in the model. So here's some data for albumin. It's listed by group. These group names aren't all that informative. If you scroll all the way down to the bottom of the page, you'll find that group difference table that we were just looking at earlier on the previous page. So it's carried along as a reference. But again, what's important here is we see that the experimental variable is the compounds. And on the left-hand side, we have some controls for graphing. And one of the ones that's used, I think, most frequently is to change how the graph is generated. And we for this case, because these were compound treatments, we'll look at the graphs based on the compound treatment. So we go back up to the top again to this data for albumin, and now it's a little clearer that which of the groups was which compound. We can click on a particular legend entry, and it will highlight the points for that legend. So it's easy to make those kinds of correlations. We can also just hover over any of these curves, and it will show us, for that particular curve. For example, this one was a combination of LPS and trovafloxacin treatment. And this one was no compound, our control treatment. So we have what looks like some good data here. We have some compounds that appear to were probably toxic and completely inhibited the albumin secretion. Those same compounds show up in the BUN plots as a decline in BUN over time. We have one treatment here, just LPS alone, that kind of has an intermediate level of BUN over time. We have this percent recovery data that I mentioned. So looking at caffeine over the course of the experiment seems to be maintained as it is flowed through the devices. We have LDH data, and again, we can hover over here. We can see that LPS plus trovafloxacin and trovafloxacin alone both exhibited an LDH release starting maybe around day 3 up to day 8. And that tolcapone from the very beginning started leaking LDH and looked pretty steadily all the way down until day 6, by which point the cells were probably dead. And so we can see more of this percent recovery data, some viability data. So lipopolysaccharide plus trovafloxacin was toxic, as was trovafloxacin alone, and tolcapone was somewhere in the middle. And then more of the percent recovery. Pretty much all of the compounds in this particular study showed a relatively high percent recovery, so there wasn't really any particular issues with compounds binding in the study. So we go all the way back up to the top on this Analyze button. We can then go and look at reproducibility to get in a sense of, for all of these measurements that were made in all of these chips, how reproducible are those values. So this is a summary over all of the assays that were run. And so you can see this particular study, the reproducibility was pretty good. We can pick something like albumin, and we can get more detail about the albumin reproducibility. We see the actual data. There were 3 chips run with the same treatment, and they look very reproducible. We calculate CVs at every time point, and so except for the very first time point where the output was near 0, the CVs all look really good. So we use 2 different metrics for reproducibility. One is the intraclass correlation coefficient. And the intraclass correlation coefficient is like most correlation coefficients. It ranges between -1 and 1, and a correlation coefficient of 1 is essentially perfect correlation, and a correlation coefficient of 0 means there's no correlation. And so if the intraclass correlation coefficients are very good here, very high. And so in the determination of the reproducibility, we first look to the intraclass correlation coefficient, and if that's acceptable, we classify based on that. Otherwise, we use the CV. We can go to the other end of the spectrum in terms of reproducibility, and we can pick out for these Presto Blue assays, say this percent control measurement. And we see that there was just one measurement made on day 10. And there, again, there's 3 replicates. And you can see the points actually don't look all that bad. The CV for these 3 data points was 15.07. That's just over our threshold of 15 for classifying an assay as poor. And so this poor classification really should be considered a flag, and it's worthwhile for the poor classification to go and look at the results, because, as in this case, it really almost made it into our classification as acceptable. So it's really still pretty good. So it's easy to come in and get an overview of the reproducibility, and then for individual assays, go in and look in detail at all of the values that went into the determination of reproducibility. The next thing I'd like to show you is our power analysis function. I think this is really very useful for study design. We have some information here. I'm going to hide that, but I would encourage you to read that if you're going to make use of the function. Start by selecting an assay for which you want to calculate power analysis. And so I'll just stick with albumin for now. And if I pick albumin, I can see all the albumin data, as we were looking at it earlier, plotted based on the compounds that were used. And I have a choice now of doing 1 sample or 2 sample power analysis. Since we have a no compound control here, we can use the two sample power analysis function. If we didn't have a control, we would have to use the 1 sample power analysis function. I won't show that today, but I would encourage you to take a look at that. And then we pick, we'll pick a compound treatment. And so we can evaluate the power of this particular assay treated with this particular compound. So we come down and we have p-values calculated as well as the power for the data that was collected. And we can see that on day 2 that the power was already up to 0.96, which is well over the 0.8 threshold that many people like to use in power analysis. But what I really wanted to show you were these power curves that give you the ability to predict how many samples, what sample size you need in order to achieve a particular power value. And the utility in that is, you know, if you look at these curves, you can hover on these curves, and if your desired threshold is, say, 0.8, you can see here, for example, that on day 2, you achieve a power greater than 0.8 with 2.4 samples. So if you wanted to run a 2-day assay, maybe you're going to run 3 samples in order to have enough. If you go 3 days, you can get by probably just about with 2 samples, or, you know, if you go out farther, of course, you can get by with fewer samples. But if you want to, you know, wanted to run this assay in a single day, you need roughly 7 samples in order to be able to achieve a power of 0.8. So it's a very useful tool for just calculating how many, what's an optimal sampling for a particular experimental protocol. So I'm going to go to, so far we've been looking at a single study and data from a single study. And what I want to show you now is how you can compare across studies. And so we have a tool we call the Graphing and Reproducibility tool. And this allows us to come in and start by selecting a model. And I'm going to select the same model we were just looking at, which is this SQL-Cell 1.5 liver model. But Texas A&M also ran this SQL-Cell microplate-based liver model. So essentially it was building the SQL-Cell model, but building it in a microplate and running that as a 2D model versus the 3D model. And so this was work done by Yvonne Rusin's group at Texas A&M. And we'll look at albumin, urea, and LDH as readouts, and I'll just select all of the compounds. And then we can go up here and we can look at the plots for these 2 different. So we're going to be looking at 2D data and 3D data from essentially the same model components, the same cells, but in different environments. In the 3D environment in a chip, and in the 2D environment in a microplate. And so you'll see in the legend, you have, again, we have these ID numbers that are the group numbers, and then they say effluent or media. So the effluent ones are the ones that come from the chips with the microfluidic chips, and the media ones come from the plates. We can again come down and switch to view by compound, and then have more informative legend to work with here in terms of the compounds. And so, you know, again, we have some compounds that are inhibiting albumin, probably through toxicity, and then we have some compounds that aren't. One of the features in this sidebar that I just wanted to show you quickly is the ability to filter out items that are not of interest, or filter to what you're specifically interested in. And so to simplify the graphs, I'm just going to go and pick a couple of compounds. Do this bar. And then we can have a graph that's a little easier to look at rather than having all of the compounds and all of the replicates of the compounds. And so here we have a no compound in effluent and media. And so as I said before, we can click on these. So there's our no compound on the chip, and here's no compound in the 2D model. And then we can look at pioglitazone in the chip and pioglitazone in the 2D model. And then tolcapone in the chip and tolcapone in the model. So in both cases, tolcapone is toxic, but you see that in the case of the 2D model that the no compound production of albumin is much lower than it is in the 3D model. And that's something that's been reported in many times. And so it's not, a particularly novel finding, but it is the sort of thing that you can easily assess in the microphysiology database, looking at models and comparing across different model types like 2D models versus 3D models. So I want to show you a couple of new features. All of these features, all of these features have been, I think I have probably demonstrated before, although I imagine there's some new people here who hadn't seen one of these demonstrations or participated in any of the webinars. What I wanted to show you is a new feature we have for managing data from cytokine panels. And so we have a study here that was looking at cytokines released from a liver model as part of a COVID-19 project. And you can see here again in this differences table that there's 4 treatments, and here the difference in the treatments isn't compounds, but it's in the media that was used. So there's late metabolic syndrome media and non-normal fasting media. So there's 2 media types and then 2 additional treatments. So if we go to the study summary, so this particular cytokine panel has 45 cytokines, and as you can imagine, it might seem like it would be tedious to enter 45 cytokines individually into the MPS database. And so what we did is we implemented a one-click load this panel option when you're creating a cytokine study that allows you to just enter all 45 of these cytokines literally in one click. And then we have all the data for all the cytokines in the graphs below. We have a new chart type option that allows you to switch to bar charts, which for this type of data is perhaps a little bit easier to see the differences from 2 to 4 to 6 days of the cytokine release from the model under the 4 different conditions. So it's a relatively new feature. I think will be of a lot of use. There's a lot of work being done these days on cytokines, and I think it's going to be well received. Then I wanted to show you, we have also the ability to handle omics data, and I wanted to show you a little bit of what that looks like. So one of the things you'll see on these tables is this column visibility button. And the things in white here are some additional features that can be added into the table. And in this particular case, what I want to add in are the omic data points. So we can see the studies for which there are omic data points. And in particular, I'm going to pick this particular study. And so like all studies, again, we have this difference table and it shows what the differences are. And here there's quite a few more differences in terms of the compound treatments, and the cells that were used varied somewhat. And there's some differences in the models. There's the 2D 384 well kidney model and the microfluidic kidney proximal tubule model. And so there's data for both of these. That was TempoSeq data that was collected by Texas A&M. And we have two different ways of looking at the results from that. One is looking at differential expression. And so this is differential expression that was calculated by Texas A&M and uploaded into the database, and then can be viewed as volcano plots. So here, for example, is a volcano plot for one of the differential expression, calculated differential expression data sets. We can pick a couple more and add some additional graphs. We have some controls here. For example, we could change the log two-fold change threshold to be 2, which means a 4-fold change. And then it's a little bit easier to see the differences, for example, from comparison to comparison in terms of the differential expression. We can also show all of the individual data points, including the ones that were not significant and aren't shown currently in this particular plot, so that the data is all there and that it's been properly filtered. So that's one way of looking at the data. And then the other way is we also capture the normalized expression values from the study. And so here again are all of the conditions that were used, and we can generate an expression matrix, which takes a little bit of time because there's a lot of data points. There's over 300,000 data points for quite a number of samples here. And so we generate a heat map. So it's a clustered heat map. And there's a lot of controls here in the heat map. We can control the clustering with some simple sliders. It's all very interactive. We can adjust the opacity or the contrast of the heat map. We can do things like change or subselect the expression, the gene expression based on variance across the rows. And so here we identify the genes that have the highest degree of variation from sample to sample. And we can arrange the genes alphabetically. So there's a lot of ability to control the heat map. We can select a particular region, a particular cluster, get a list of the genes in that cluster. We can send these genes off to Enricher, which then gives us information about transcription and pathways and drugs. So it's really very interactive and very easy to use. Sorry. So that was most of it. One last thing on the MPS database, and that is we've developed this COVID-19 portal. And so in general, we believe that these portals can be very useful in terms of providing users a quick way to access information about diseases and models and all of that. And so we have summarized in this one portal, we have general information like government resources and literature sources, information about disease biology that's linked directly from here, various kinds of experimental models, commercial models, research models, and components, as we were talking about earlier, components with that are used in constructing all of these models. And so this, to us, seems like it will become a very useful way to bring together, easier access to information for people who are building and working with models. So that's all for the tour. I just want to mention two other portals that we're working on. One is a stem cell information portal, which will provide access to stem cell sources and identify stem cell centers with information about what stem cells they have available and how to contact them and all that sort of thing, including information about literature sources, and then biology of stem cells and protocols for differentiating stem cells into various cell types. And a second one is a preclinical trial portal, which I mentioned earlier that it's becoming possible with the stem cell derived models to create a cohort of models that represents multiple patients that could be used to run a preclinical trial in a chip format, for example. And what will be really important is to be able to access those cohorts of iPS-derived cells and MPS models. So this is just a reminder that we like feedback and hope you'll give us feedback when you register and use the MPS database. And then we're always willing to give additional training. So I went over a lot of stuff quickly today. I'm sure that you're going to come away with questions. We have our online webinars, but please contact us and we will give you some more one-on-one help. This is the team that has been working on and developing the MPS database and in various roles. And then I just had a few topics to perhaps stimulate some discussion. We'd be very interested in working with any of you to add data from experimental models that are either in development or being used at the NIDDK. We'd certainly be interested, when we're always looking to add additional data from animal models and especially identify new sources of clinical trials data. And so we'd certainly be interested in discussing that if people have ideas about that. And then just as a general question, what would you all like to see in the MPS database? What could we do to make it more useful to you? So with that, I'll leave it to questions and answer session.
Okay, thank you, Dr. Gough. I have one, probably it's a question from the chat. It's from Anita. Have all of the portal that you are working on have been registered with our ID initiative?
I'm not sure I understand the question.
Let me see. Oh.
So registered, you mean?
Yeah, research, RRIDs are research resource identifiers. It's one of the services that dkNET helps to provide. And these are unique identifiers that that can be used to reference individual databases such as yourselves. In the biomedical literature, they're used by journals as a referencing system. And they're also given to key reagents such as cell lines, antibodies, software tools, organisms, biosamples. And so I don't know if you're familiar with the FAIR data principles, but the FAIR data principles really promote the use of these unique identifiers because they are computable and linkable. So
So I am familiar with the FAIR principles, but the short answer is no, we don't have an ID, but certainly would be interested and
Yep.
we'll follow up after this webinar on
I was already
seeing that
looking
we
it
can
up
get that in place.
to see if you were there. But I did want to say that it's a really fantastic resource that you've put together. And when you say adding data from experimental models in development or use, do scientists from the community, are they able to put data in, or is this something curated that you yourselves do? I mean, how
No,
does it get?
yeah, absolutely. The preferred approach is to have the scientists put it in themselves until we train scientists. We're currently working with 15 different groups from NCATs that are funded on microphysiology systems development, organs on chips, and disease model development, and all of those. And so our general approach is, as I try to portray in this presentation, we've worked hard on developing a streamlined approach to getting data into the database and creating studies and all of that. And so we train individuals on that, and then they upload the data themselves. So we'd be, if there's people within NIDDK that have models or using or something, would like to make use of the MPS database resource, they should contact us and we can give them some help in getting started with creating and uploading data. And there's a little bit of setup we have to do in advance for each group that's going to be contributing data. We have to establish a center for them because that's how we track them. And that's something that we do at the very beginning with the group. And then once their center is created, they can operate more or less independently, but we still give them training as needed.
Okay, there's another question from the audience. The audience want to ask, so you mentioned on the one of question, which result from the organ model are predictive of a specific clinical outcome, and how reliable is the prediction? Could you show us again?
So I can't show you that per se. So what I did say was that we're currently in the process of developing a tool to do exactly that, to allow you to take the data from your experimental model and use that to evaluate its concordance with clinical measurements. And so that's a work in progress at this point in time, but it's high on our list, and it's something we've been working a lot on, and should have a solution for that in the next few months.
Thank you. And then I think there's one question. It's during your presentation. It's probably it's a minor question. So I think the audience asked, when you click a view cell sample, it seems like have username is required. So it's open to anyone that can use this platform, or they need to pay a fee to see a certain data.
All right. So there's two things. One is many things are accessible without registering on the MPS database, but registration only takes like a minute. You just have to sort of enter your email address. We keep that information all private so nobody else sees it, so you won't get spammed from registering on the MPS database. And we'd highly encourage you to do that. There is some information that's only accessible after you have registered. And then there is a lot of information that's in the database that's hasn't been released publicly yet, that individual scientists are working on studies and creating studies and uploading data. And at the end of that process, once they're completed that study and they've got all the data in the database and they sign off on the study, then the study is typically made accessible. So at this point in time, if you go into the MPS database and register on the MPS database, you'll see a lot of public studies. I can't remember how many we have, but there's a couple hundred, I think. And that's probably about half of the studies that are in the database right now, but the other ones are still sort of in process and will be released once they've been completed. So it's definitely useful, I think, overall to register because you'll get the maximum access to information in the database, and it doesn't cost anything, and it's kept private. Nobody will see your name or email address in the MPS database, unless you list yourself as a contact for a model or something like that, which those are the few cases in which that a email address is exposed, but that's usually because the person wants it exposed so that they can be contacted if somebody's interested in their model.
Thank you. There are also some questions in the chat. I think previous one is RID for Microphysiology System Database. And then I think Anita also asked, how do people cite the models that are listed in MPS database?
So it's with respect to models, it's kind of the other way around, that in the model, people typically will reference their publication for the model, of the demonstration of the model, what the model can do and all of that. And so there's a place under models to link publications that links to PubMed directly and allows you to access those publications for the model. For the studies, we do have the ability to create a set of studies, and I didn't show that feature, but what you can do in terms of creating a set of studies is you can take any combination of study data that you've generated in the database, and you can combine that under a single URL that gets assigned as a unique locator for that combination of study data. And so people, when they publish, they will take all of the data that they used in their publication, and they will combine it into a study set, get that URL for that study set, and then reference that URL in the publication. And then that allows people who are reading the publication to easily and quickly just click the link and end up at the MPS database and have all of that data in the MPS database plotted. And in addition, they can download the data in a CSV file if they want.